Reversing the effects of sickle cell retinopathy using a gene circuit to synthetically dilate blood vessels around the retina. And we are from Andover High School's Bodybuilder Club. Sickle cell disease, or SCD, is a type of blood disorder that affects the hemoglobin in red blood cells, which is responsible for delivering oxygen to cells throughout the body. This autosomal recessive disorder is caused by mutation in the HBB gene that produces an abnormal version of hemoglobin's beta globin subunit called HBS. This mutation causes red blood cells to become crescent, or as most people know them, sickled. Sickle cell retinopathy, or SCR, is a complication of SCD that affects the retina and choroid blood vessels in the eye. The abnormal shape of sickled red blood cells causes vascular occlusion or blockages in blood vessels, which leads to reduced blood flow and lack of oxygen in the tissues of the eye. This can cause a range of symptoms, including vision loss, blurred or disturbed vision, dark spots, floaters, and sudden blindness. Unfortunately, the current treatments for SCR are limited, and there is a need for new effective therapies. Our proposed solution involves the creation of a gene circuit, which generally is a set of genetic components such as promoters, coding sequences, and terminators that interact to produce a specific output in response to a particular input, or in our case, set of inputs. Our gene circuit is designed to specifically address the problem of sickle cell retinopathy. The circuit is engineered to detect the presence of sickled red blood cells and hypoxia around the retina and then produce nitric oxide to dilate blood vessels and allow for sickle cells to pass through and still deliver oxygen to the eye. So basically, it works as a biological stent. Nitric oxide is a potent vasodilator, therefore when produced in blood vessels around the retina, it will relax the smooth muscle and allow oxygen to flow, even sickled cells, into the retina and choroid. One of the advantages of our gene circuit is that over current treatments for SCR is that it is targeted specifically to blood vessels around the retina. And therefore, we are addressing the root cause instead of just repairing damaged retina, which will help get the most long-term effects and therefore the most effective treatment. Our circuit functions as an AND gate, which is formed by the combination of two inputs to produce an output. The first input is sickled cells, which is detected by the sickled cell sensor promoter and encoding sequence. And the second input is hypoxia, which is detected by the HB sensor promoter and encoding sequence. After sensing these two inputs, the nitric oxide synthase gene will be activated, which will produce nitric oxide synthase in the blood vessel, which will induce vasodilation and increase blood flow to the retina. Once the gene circuit is injected, it'll begin to detect sickle cells and low oxygen levels right away, and the nitric oxide will get produced and diffused into the surrounding tissues. For a plasmid, here is a diagram of our AND gate. The CMB promoter initiates transcription of every component of the circuit. The HBB sensor promoter and coding sequence are used to detect low oxygen levels in red blood cells. The sensor works the way it does because it contains a regulatory element known as the hypoxia response element, or HRE, which is able to detect low oxygen levels in the surrounding environment. Sickle cell sensor promoter and coding sequence detect the presence of sickled red blood cells in the blood vessels, and the sensor works the way it does because it is able to detect the abnormal beta globin subunit HBS that is only present in patients with sickle cell disease. Once these two inputs are detected, the nitric oxide synthase gene encoded by the nitric oxide synthase coding sequence will be produced in response to activation by the transcription factors. We have also added in the HIF1 alpha protein coding gene and the HBB sensor coding sequence. When oxygen levels are low, such as in the eye due to a blood vessel blockage, the HIF1 alpha is stabilized and activates genes that promote adaptation to hypoxic conditions. In this gene circuit, the HIF1 alpha protein activates expression of the nitric oxide synthase gene in response to hypoxia. We have also included the NRE and HRE regulatory elements to regulate gene expression. And finally, we have the bovine growth hormone polyadrenaline signal, which is a commonly used terminator that we will use to ensure proper termination of transcription. Thank you for listening.